Welcome to the chemistry lecture on urea and serum creatinine. I won't be reading through the objectives, but will instead begin today with sources of nitrogen on slide 6. Nitrogen is an important element that is necessary for life. It is an essential component of several metabolic compounds such as proteins and amino acids and nucleic acids. The metabolism of any substrate will ultimately result in the production of waste products. For example, the complete metabolism of glucose results in producing the waste products carbon dioxide and water. The metabolism of nitrogenous compounds results in production of waste products that contain nitrogen. There are 4 primary nitrogenous waste products that we will be discussing. They include uric acid, ammonia, urea, and creatinine. This lesson will cover the last 3. Uric acid will be covered next week. Proteins in the body come from 2 sources. One is exogenous, or from dietary sources, and the other is endogenous, from normal cell turnover. In both cases, metabolism of these proteins contributes to the amino acid pool. Amino acids can be metabolized directly for energy, which will result in removal of the amine group, a process called deamination. Deamination is a hydrolytic reaction that requires addition of water. The NH2, or amine group, that is removed gains the 2 hydrogens from water, thus becoming NH4, or an ammonium ion, or ammonia. Ammonia is highly toxic to the body, so that only very low levels are tolerated. That being the case, ammonia is converted to urea in the liver. Urea is also toxic, but much less than ammonia. Amino acids are also necessary for the synthesis of creatine, which can become phosphorylated to phosphocreatine, a storage form of energy in the muscles. When creatine is metabolized, it becomes creatinine, which is ultimately excreted in the urine. Amino acids are also necessary for production of nucleic acids, such as DNA. Nucleic acids can be divided into 2 groups, purines and pyrimidines. You should recall that there are 4 nitrogenous bases that comprise the nucleotides and are adenine, thymine, guanine, and cytosine. Adenine and guanine are purines, while thymine and cytosine are pyrimidines. When nucleic acids are metabolized, such as by normal cell turnover, the purines are metabolized to uric acid, while the pyrimidines are metabolized to ammonia. Though there are several sources of nitrogen in the body, remember that the major source is proteins. As you can see on this slide, urea comprises the highest proportion of non-protein nitrogenous compounds in the body for 2 simple reasons. 1, urea is synthesized directly from ammonia, and 2, ammonia is derived from protein catabolism. Uric acid and amino acids come in second at 20% each, followed by creatinine, creatine, and ammonia. It's not surprising that ammonia is present in such low quantities due to its high toxicity. The human body excretes excess nitrogen through production of nitrogenous wastes. Some of these are processed further in the liver, such as ammonia, but ultimately they are filtered by the kidneys and excreted in the urine. Of course, as I've mentioned previously, urea is synthesized in the liver. Although urea has a simple molecular structure, it has a rather complex metabolic pathway. Fortunately, it is not necessary to learn the specifics of this pathway. However, if you study it, you will quickly see that it is very similar to Krebs' cycle in the glycolytic pathway, and for good reason. The scientist that identified and described Krebs' cycle, Hans Krebs, also discovered and described the urea cycle. This slide illustrates the hydrolytic reaction required to deaminate an amino acid. The elements are colored so that if you follow the reaction, you can see exactly what happens. The oxygen from water replaces the amine group on the amino acid, resulting in an alpha keto acid. Meanwhile, the two hydrogens from the same molecule of water combine with the amine group that was removed to form an ammonium ion. It is very important to remember that ammonia is highly toxic to the body. Thus, the levels of ammonia must be kept low in circulation. In addition, ammonia can influence the acid-base balance in the body. That is why it is not uncommon for people in end-stage liver disease to develop acid-base disorders, primarily metabolic alkalosis. This happens in end-stage liver disease due to the liver's inability to process ammonia to urea. Under normal circumstances, greater than 90% of urea is filtered freely in the glomerulus. However, a large proportion, between 40 to 70%, is passively reabsorbed in the DCT and collecting tubules. Several factors influence the amount of urea that is reabsorbed, but the main one is how hydrated the body is. You see, urea helps with water reabsorption by contributing to the osmotic pressure in the medulla of the kidneys. Although urea is freely filtered, it can also be secreted, but usually only seen in renal disease. Incidentally, a small amount of urea is also excreted through the gastrointestinal tract and in the sweat. So far, we've only talked about urea. Now it's time to talk about blood urea nitrogen, or BUN. There's really nothing complicated about BUN. It is simply determining the nitrogen content of urea. So, here I've depicted one molecule of urea, which is composed of one carbon, double bonded to one oxygen, and two amines, each bound to the same carbon. So, the molecular formula is CONH2 with a molecular weight of 60, since nitrogen equals 14, hydrogen equals 1, carbon equals 12, and oxygen equals 16. The weight of nitrogen in one molecule of urea is 28. So if you take a BUN value and multiply it by 60 divided by 28, you get the equivalent urea concentration, or BUN in milligrams per deciliter times 2.14 equals urea in milligrams per deciliter. Now, everyone else in the world simply measures urea and reports out urea concentration. However, in the US, we measure urea and report out BUN. Why, you might ask? It simply comes down to habit. You see, in the old days, we used to measure all of the non-protein nitrogenous compounds and calculate an index fraction for each based on the nitrogen content. We no longer do that. However, the practice of reporting urea nitrogen stuck, as that is the value physicians are trained to interpret. When it comes to abnormal levels of urea, we really only care about increases. Increases in urea can either be due to uremia or azotemia. Uremia, as in uremic syndrome, is an increase of urea circulation due to renal disease that causes decreased glomerular filtration. Azotemia is a bit broader in its definition. It can be due to either an increase in urea or an overall increase in nitrogen. We'll take a look at both. We'll start with azotemia. The causes of this condition can be either renal, pre-renal, or post-renal. Pre-renal causes can be due to reduced blood flow, low blood pressure, or both. For example, congestive heart failure, shock, acute hemorrhage, and even dehydration. When there is a reduced blood flow through the kidneys, or reduced pressure, the glomerular filtration rate drops, and wastes are not filtered as effectively. Other potential causes include diet, an increased nitrogen intake, increased protein metabolism, such as malignancies causing increased cell turnover, and fever and illnesses due to increased metabolism caused by increased body heat. Renal causes of azotemia can include both acute and chronic renal failure and glomerulonephritis. Post-renal causes are due to some sort of urinary tract obstruction, such as renal calculi, or kidney stones, enlarged prostate, decreased urine output, tumors, or physical blockage of ureters or urethra, and infection, bacteria converting urea back to ammonia with reabsorption into the blood. Testing for BUN in serum or plasma samples is primarily done to help assess kidney function. As mentioned earlier, we are only really interested in increased values of urea, since this may indicate a problem. Interpreting BUN values is not without its problems, though. There are several factors that can directly influence BUN, including how hydrated the patient is and the degree of protein catabolism, which is highly influenced by diet. Ultimately, we are trying to determine if renal impairment exists or not, and due to external factors that can affect BUN values, this test should not be used exclusively to determine that. Other contributing factors that can cause an increased BUN include intestinal obstruction due to increased absorption, cardiac failure due to decreased blood flow through the kidneys resulting in low glomerular filtration, dehydration causing more urea to be reabsorbed in the medulla to help concentrate the urine and conserve water, and diet if there is a higher protein and/or nucleic acid intake. Factors that can contribute to a decreased BUN include diet if there is a lower protein and/or nucleic acid intake, severe or end-stage liver disease due to the inability of the liver to convert ammonia to urea, severe vomiting and/or diarrhea due to a decreased absorption of protein and nucleic acids, and pregnancy due to nutrients being diverted to the developing fetus. While urea is widely associated with kidney function, it does not It does share a connection to liver function as well, that being the conversion of ammonia to urea. However, BUN is not used to assess liver function, and for good reason. Since ammonia is so toxic, the liver retains its ability to convert it to urea, even at the expense of other functions. Therefore, when ammonia levels rise and urea production decreases, it is a sign of extreme liver damage. The normal range for BUN in serum and plasma is generally 7 to 18 milligrams per deciliter and 7 to 16 grams per 24-hour volume for urine. Now, let's say the average urine volume per 24 hours is 1,200 milliliters. That is the same as 12 deciliters, since there are 100 milliliters per 1 deciliter. Now that we've settled on that, let's take the lower reference limit, 7 grams, and convert that to milligrams per deciliter. We'll start with 7 grams per 12 deciliters, which equals 0.583 grams per deciliter. Multiply that by 1,000 to convert grams to milligrams, and we get 583 milligrams per deciliter. If we do the same to the upper reference limit, we get 16 grams divided by 12 deciliters equals 1.333 grams per deciliter, or 1,333 milligrams per deciliter. So as you can see, if we receive a 24-hour urine collection, we need to measure the total volume collected, measure the urea from a well-mixed aliquot, and divide the urea value by the urine volume multiplied by 1,000 to get the urea concentration in milligrams per deciliter. One of the very first methods developed for measuring urea was the diacetyl method. It was a simple reaction that involved addition of the reagent diacetyl to the patient sample. Diacetyl reacts with urea to form the yellow compound diazine, which can be measured spectrophotometrically. There are, however, several disadvantages to using this method. First, there are non-specific reactions that occur with other substances present in the sample, causing falsely elevated results. Second, the reaction does not follow Beer's law very well, necessitating the use of many standards. And last but not least, diacetyl is a noxious and dangerous reagent to use. So, not surprisingly, it is no longer used. Next up are the enzymatic methods. Most modern-day methods for measuring BUN require use of the enzyme urease. This enzyme reacts with urea, converting it to ammonium ions. After that, though, each method will be unique for the manufacturer of the test. Take, for example, Berthelot's reaction. This method requires the addition of an alkali following the initial reaction with urease. The alkali will convert the ammonium ions to ammonia. Ammonia can then react with a phenolic compound and sodium hypochlorite in the presence of the catalyst sodium nitroprusside to form a colored compound. The exact color of the compound depends on the phenolic compound used in the reaction. In any case, the amount of color produced is directly related to the concentration of urea in the sample. Another option for measurement of BUN is to incorporate the use of a second enzyme, glutamine dehydrogenase. In this case, ammonia reacts with alpha-ketoglutarate and NADH in the presence of glutamine dehydrogenase to produce glutamate and NAD. As we can see in this reaction, NADH is oxidized to NAD, and this oxidation reaction can be measured at 340 nanometers. There is also an enzymatic conductivity method that can be used. This method is based on measuring the increase in electrical conductivity as urea is converted to ammonium ions in the first reaction. The rate of increase in conductivity is proportional to the concentration of urea in the sample. Creatine, not creatinine, is synthesized in the liver from 3 amino acids: arginine, glycine, and methionine. After becoming phosphorylated by ATP and the enzyme creatinine kinase, it is stored in the muscle tissue for energy. In terms of its metabolism, there are 2 pathways it can take. The major pathway is conversion of phosphocreatine to creatinine. The minor pathway is conversion of creatine directly to creatinine. Both pathways occur in the muscles at a relatively constant rate of 2% per day. Creatinine is the metabolic byproduct of creatine and phosphocreatine metabolism. It is inert and therefore will not react with any other substances found in the urine. It is produced at a constant rate dependent on muscle mass, but independent of diet, unlike urea. It is excreted in the urine under normal circumstances by glomerular filtration without any significant reabsorption, though it can also be secreted directly into the tubules. More on that later, though. Serum and plasma can be measured. The normal range is 0.2 to 1.5 milligrams per deciliter for both males and females. The normal range for urine, though, is gender specific. For males, the range is 20 to 26 milligrams per kilogram of body weight per 24-hour volume. And for females, the range is 14 to 22 milligrams per kilogram of body weight per 24-hour volume. Now, let's see how to convert a value into milligrams per deciliter. First, let's pick a value for a male patient. How about 23 milligrams per kilogram? And let's say this patient weighs 82 kilograms. 1 kilogram equals 2.2 pounds, so 82 kilograms is about 180 pounds. And the 24-hour volume is 1,200 milliliters, or 12 deciliters. 23 times 82 equals 1886 milligrams per 24-hour volume. 1,886 milligrams divided by 12 deciliters equals 157.2 milligrams per deciliter. So, I mentioned earlier that under normal circumstances, creatinine is filtered by the glomerulus without any significant reabsorption. It can, however, be secreted directly into the tubules when the glomerular filtration rate decreases. Blood creatinine levels do not begin to rise until the GFR decreases by around 30 to 40%, which translates to moderate kidney damage. That means that if we use serum creatinine levels alone to assess kidney function, we wouldn't identify someone with kidney disease until they were in stage 2 or stage 3. Stage 4 is end-stage disease. This is another reason why multiple tests are required to adequately assess organ system function. The classic method for measuring creatinine, and still the most common method, is the Jaffe reaction. This method requires reacting creatinine with alkaline picrate to produce a red-yellow colored compound that can be measured spectrophotometrically. There are, of course, substances that can interfere with the assay. They include glucose, protein, ascorbic acid, or vitamin C, ketones, and cephalosporins. The answer to proteins is to either filter the specimen prior to reaction, which is what automated instruments do, or prepare a protein-free filtrate. The other interfering substances can be removed by the addition of Lloyd's reagent, or sodium aluminum silicate. This reagent will bind with the interfering substances and prevent them from reacting with alkaline picrate. Since we also routinely test urine using this method, and urine contains a lot of creatinine, we must first perform a dilution before testing to keep values in linearity. The upper reference limit for serum creatinine is 1.5 milligrams per deciliter, and the upper reference limit for urine is 175 milligrams per deciliter. That means, at minimum, a 1 to 175 dilution should be made. We typically make a 1 to 200 dilution to be safe. The Jaffe reaction can be performed as an endpoint or multi-point, or kinetic assay. You will be performing this test as an endpoint reaction in the lab, but chemistry analyzers perform this as a multi-point assay. The advantage of the kinetic assay is illustrated in this slide. In some instances, there may be a long lag time where interfering substances are being removed, followed by the reaction with creatinine. In this instance, only the measurements between A and B will be used to determine the slope of the reaction. The last method is enzymatic and requires creatininase. This enzyme catalyzes the conversion of creatinine to N-methylhydantoin and an ammonium ion. The ammonium ions produced can then be measured with either an ISE to measure electrical conductivity or reacted with an indicator dye such as bromophenol blue. The last topic to cover is the BUN creatinine ratio. The normal ratio is anywhere between 10 to 20 to 1. There are, however, conditions that can affect this ratio. For instance, both glomerular and tubular disease will cause increases in both the BUN and creatinine values, but they will be proportional increases. Because of this, the BUN creatinine ratio will remain normal. Pre-renal azotemia and a high protein intake typically cause an increase in the BUN, but no increase of creatinine. This results in a high BUN creatinine ratio. We can also see a high ratio, but along with an elevated creatinine, an increase in both, but not a proportional increase, with conditions such as post-renal urinary obstruction and pre-renal azotemia with renal disease. Low ratios resulting from a decreased BUN and a normal creatinine are typically seen with low protein intake or starvation, liver disease, following diarrhea or vomiting, and following renal dialysis.